Imagine a world where we could edit out diseases from our DNA. Sounds like science fiction, right? Well, it's not. Thanks to gene editing technologies like the CRISPR-Cas9, we're on the brink of a medical revolution. Whether it's curing genetic disorders, enhancing crops, or even fighting cancer, gene editing holds massive potential. But it also raises some serious ethical questions. Today, we'll dive into how gene editing works, what it can do, and the debates around it. So, let's get started. Gene editing isn't new. In fact, scientists have been trying to alter DNA for decades. But the game changer came in 2012 with the discovery of the CRISPR-Cas9. CRISPR, short for Clustered Regularly Interspaced Short Palindromic Repeats, is a natural tool bacteria use to defend themselves from viruses. Think of it like a genetic cut-and-paste function. CRISPR can find specific sequences of DNA, cut them out, and even replace them with a new piece. And Cas9 is the enzyme that makes the actual cut. But why is this a big deal? Well, before CRISPR, gene editing was slow, expensive, and much less precise. CRISPR is cheap, fast, and accurate, making it a powerful tool for scientists and doctors alike. Have you ever imagined editing your own DNA to remove a disease? Alright, so how does CRISPR-Cas9 actually work? Let's break it down. First, CRISPR needs to find its target. It's like a GPS system for DNA. Scientists design a piece of RNA that matches the DNA sequence they want to edit. This guide RNA then directs the Cas9 enzyme to the exact spot on the DNA. Once CRISPR finds the right location, Cas9 acts like a pair of molecular scissors, cutting the DNA at that specific spot. And this is where the magic happens. After the cut is made, the cell's natural repair mechanisms kick in. Scientists can either disable a gene to stop it from working, or insert a new piece of DNA to replace the faulty one. Think of it sort of like editing a typo in a document, but instead of fixing a word, you're fixing a gene. This makes me curious, what would you edit if you had access to CRISPR? Drop your thoughts in the comments down below. Alright, now let's talk about how CRISPR and other gene editing technologies are actually being used in the real world. One of the most promising uses of CRISPR is in its curing genetic disorders like sickle cell anemia and cystic fibrosis. Scientists are using gene editing to correct the faulty genes responsible for these conditions. And guess what? We've already seen successful treatments in clinical trials. CRISPR is also being used to fight cancer. Researchers are editing immune cells to make them more effective at recognizing and attacking cancer cells, giving patients a better shot at survival. But it's not just about human health. CRISPR is also revolutionizing agriculture. Scientists are creating disease-resistant crops, enhancing nutritional content, and even speeding up the growth of certain plants. I remember reading about how CRISPR was used to edit mosquito genes to reduce the spread of malaria. So just imagine wiping out a disease completely by editing a mosquito's DNA. I don't know, what do you think? Should we use gene editing to alter nature? Go ahead and let me know your thoughts in the comments below. Anyways, as exciting as gene editing is, it's really not without its concerns. One issue with CRISPR is that it doesn't always hit its target perfectly. Sometimes it can make unintended cuts in the DNA, which could potentially cause new problems. But scientists are working hard to make CRISPR more precise. Another big ethical concern is the idea of designer babies. Should we use gene editing to enhance traits like intelligence or physical appearance? I mean, while this might sound futuristic and kinda cool, it does raise some serious ethical questions about fairness and inequality. And then there's also the debate around gene drives. This is when scientists edit the DNA of a species in a way that forces the new genes to spread through an entire population, potentially altering ecosystems. While this could help with problems like invasive species, it could also have unintended consequences. What's your take on these ethical concerns? Should we regulate gene editing, or should we push forward? Go ahead and let us know in the comments. So, what does the future hold for gene editing? Well, in the future, we could see cures for more genetic diseases, personalized medicine tailored to your DNA, and even advancements in organ regeneration. Just imagine getting a new heart grown from your own cells. But with these advances, the ethical debates will only intensify. Who should be having access to gene editing? How will it be regulated? And how do we ensure it's used responsibly? So, there you have it. This is how CRISPR and other gene editing technologies are revolutionizing medicine. From curing diseases to enhancing crops, the potential is endless, but the ethical questions are just as important. If you found this breakdown helpful, make sure to smash that subscribe and ring the bell for more tech explainers from TechWkly. And remember, with great power comes great responsibility, even when it comes to editing genes. Thanks everyone for watching. For more content like this, remember to like, subscribe, and hit the notifications bell.